talking about is called a CRISPR-Cas9 gene drive system targeting female reproduction in the malaria, mosquito vector, Anopheles gambiae. But what it's about is engineering the mosquito population to combat the spread of malaria. So malaria is an infectious disease caused by microbes called Plasmodium, which infect red blood cells. So in the short term, you can see some of the symptoms over on that image. In the short term, a lot of the symptoms are the same as the common cold or the flu, stuff like chills, fever, fatigue, headaches, vomiting, stuff like that. But in the long term, because this is an infection of blood cells, it causes anemia, which is where you don't have essentially enough red blood cells, which eventually leads to death. So in 2015, 438,000 people died this way. So worldwide, it's very common in tropical areas, which unfortunately are also often poor third world countries that don't have access to great medical supplies and such. You can see on the map it's largely found in Central Africa and also in South America and Southern Asia. But it's not just sort of an isolated problem. Worldwide, 3.2 billion people are threatened by malaria, which is close to half of the world's population. So attempts have been made to create a vaccine or a drug that will combat malaria, but so far they've only had limited success because the malaria has developed immunity to a lot of the stuff that's been created. So instead, a lot of research now is focusing on what's called vector control. Basically, malaria needs a vector, some other organisms, to spread it from human to human. So if you can control the population of that vector, you can control the spread of malaria. So that vector is mosquitoes, and specifically Anopheles gambiae is the most common vector for malaria. And the reason the malaria needs this is because it's not like malaria isn't like a cold or the flu where you can get it just through contact or by breathing on someone. It needs to directly come from the blood of one person to the blood of another. So the female mosquito will suck human blood to feed its eggs. And if it does that and it sucks blood from someone who has malaria, then it becomes infected, and anytime it bites another person, that person will also become infected. But importantly, the mosquito itself is not in any way harmed by the malarial infection. So there's no sort of evolutionary force driving the mosquitoes to develop any sort of resistance to this. So the way researchers are trying to combat this in this paper is by introducing a mutation that will reduce the fertility of mosquitoes to reduce the population. So the mutation they generated affects the ability of the mosquito's ovaries to form eggs properly, which importantly will reduce, or it'll pretty much knock out the female mosquito's ability to have children, but it won't affect male mosquitoes at all. So any male mosquito with this mutation will not be affected and will be able to spread the mutation to all of its offspring. So as you probably know from bio class, there's 2 copies of every chromosome in an animal, and therefore 2 copies of every gene, one that comes from the mother and one that comes from the father. So the researchers looked at mosquitoes with either 1 mutant copy of this gene or 2 mutant copies of this gene. Those are the homozygous and heterozygous mosquitoes. So what they found here, they're looking at the number of viable larvae that the mosquitoes were able to produce. Here at the top, you see the heterozygous mosquitoes, the ones with only 1 copy of this mutant gene. And as you see, they're able to produce, plenty of offspring. Yeah.
How do the numbers of the heterozygote compare to wild type?
They did not look at that. They were just comparing the heterozygote to the homozygote. So then when you look at the homozygote, you see pretty much 0 normal progeny were generated.
Are you sure? They didn't look at the wild type?
pretty sure.
Okay.
So importantly, if you only have one copy of this mutant gene, it doesn't completely sterilize the mosquito. You'll need 2 copies. So does anyone have any idea how this could be a problem if you're trying to introduce sterility into the wild population?
Mosquitoes are probably preying for another animal, so if you get rid of mosquitoes, you could be knocking out a source of food for another animal.
Well, that's sort of a separate issue. I'm talking specifically, how does the fact that there's a difference between the heterozygote and the homozygote cause a problem if you're trying to reduce the size of the population?
Just that is it that it's more likely that you're gonna have a heterozygote mixture, and like a homozygote would be harder to like find 2 mosquitoes that have that gene.
Right, exactly. If you release a male with this mutation into the wild and it mates with a female from the wild that doesn't have this mutation, all of their offspring will have at least one non-mutant copy of the gene. So all those offspring will be able to produce more larvae, no problem. So what they did in order to combat this is they employed something called CRISPR-Cas9. This is a tool that's really big in biotechnology. It allows researchers to do all sorts of cool genetic modification type stuff. So the way it works basically is you have this Cas9 protein here, the green one, which has a guide RNA here. And the sequence of that guide RNA corresponds to a sequence somewhere in the genome. So the guide RNA guides the Cas9 protein to that sequence, and it cuts out the sequence. So now the cell wants to repair that cut. In order to repair that cut, it needs some sort of template, and that template will be some piece of DNA that the researchers introduce. So they use this to create something called a gene drive. So the way this is normal inheritance, right? Again, you have a mutant fly and you have a mutant fly and a wild type fly. They have offspring. The offspring has one mutant chromosome and one wild type chromosome. So what the gene drive does is it produces the Cas9 protein, which cuts the wild type chromosome. And when the cell tries to repair it, the only template sequence it has is this mutated chromosome. So it effectively converts the heterozygote here into a homozygote. So now this mosquito here is going to pass on this gene to all its offspring if it's a male, and if it's a female, it'll be completely sterile. So they wanted to look at what this actually does in a population of mosquitoes. So they took 300 wild type, that's normal, not mutated mosquitoes, and 300 mutated mosquitoes, and essentially just let them reproduce for 4 generations. These red lines here are two of the actual experiments with real mosquitoes. All these gray lines in the back are just computer simulations based on how they think it'll work, and you see, the most important is obviously the real mosquito populations, and so you see that the number of the percent of mosquitoes with this mutation increased from 50% to about 75% over the course of 4 generations, and this is not expected because normally you would expect the ratio to stay at about 50%, or even possibly to decrease from there, because this is a gene that sort of decreases your chance of survival or of passing on the genes. So this shows that there is abnormal inheritance going on, which indicates that the gene drive was able to create homozygotes. Yeah.
When you say generation, do you know how long, like specifically in years or days or hours that would be?
I think it's about a couple weeks for mosquitoes. Alright. So they wanted to make sure that they're still effectively sterilizing the female mosquitoes. So again, they're counting the number here of larvae and of eggs that were produced from the from the females in this population that had the mutation. And again, it's close to 0, a little bit higher than in the previous study, or the previous example, but still it's effectively 0. They're effectively sterilizing the female mosquitoes. So this might seem a little paradoxical, right? Because they're trying to pass on this mutation from one generation to the next, but it's sterilizing the mosquitoes. Does anyone have any idea how this actually gets passed on then?
Through the male?
Exactly. So as I said before, it only sterilizes female mosquitoes. The male mosquitoes are still able to have offspring. So if you have a mutated, group of mutated mosquitoes, the females will all be sterile, and the males will all be able to produce more offspring. So just to summarize what they did, they were able to create a CRISPR-Cas9 gene drive that increases the frequency of a mutation in the population of mosquitoes. And that mutation was effectively able to sterilize female mosquitoes, which in theory over time should decrease the size of the population. So that's ultimately the idea here, is you want to be able to introduce some of these mutated mosquitoes out into the wild, and over time that'll reduce the population size by spreading these mutations throughout the population. As I said before, it can be a little difficult because we're effectively trying to spread a gene that decreases normal evolutionary fitness. So you're kind of working against regular evolutionary pressures and regular survival of the fittest type stuff. So it can be a little hard to predict how this is going to work out in an actual a much larger population over dozens and dozens of generations. So there needs to be more research into in larger populations and over longer periods of time how this works out. The other thing that can be hard to predict, though, as someone mentioned earlier, is sort of the effects that this is going to have on the rest of the environment. So ecosystems are very complicated, and every species, every population has some effects on every other population in the ecosystem. So if you're suppressing this one population of mosquitoes, you very well could you probably would be successful in decreasing the rates of malaria, but you could also have all sorts of other effects on the ability to develop agriculture in that region, or the ability for humans to live in that region. So I'm not saying that this is a line of research that we shouldn't investigate further, but it's definitely something we have to be prepared to deal with. If we end up using this ability to engineer entire populations. Any questions?
Why not just vaccinate people?
Vaccines haven't been super effective.
How could this be more effective than the current vaccinations that we have?
Because you're basically knocking out the malaria's ability to spread from person to person.
But wouldn't the vaccine just do the same? Because you said that the mosquito is just a vector, so if the mosquito bites someone who doesn't have malaria in the first place, then they can't spread it.
The malaria, there have been attempts at creating vaccines, and the malaria has developed resistance to those. So, they can't really develop a new way to spread from person to person.
So you showed what happened over the course of generations as far as gene frequency.
Yep.
But was there actually a change in the population level? Did they see that over the
They just looked at a sample from each generation to get the frequency of the mutation. They didn't actually look at the size of the population, so I'm not sure.
In that study with 300 mutated mosquitoes, did they also use females for the mutations?
They were, I believe the original 300 wild type and mutated were, roughly the same number of males and females.
I'm just thinking about what's gonna happen when like the GMO people find out about this. They're gonna flip.